Hey everyone, and welcome to The Explainer. Today we're literally diving into the very code that makes us who we are. We're going to explore this absolutely fascinating tool created by Heinrich Ferreira called the Visual Genome Browser. It's designed to map out our biology in a way you've probably never seen before. And honestly, it's a real game changer.
But first, picture this for a second. Imagine you're handed a book filled with 3 billion letters. There's no formatting at all. No spaces, no paragraphs, 0 chapters. It's just a massive, endless, linear sequence of text. How on earth do you navigate that without getting entirely lost? Well, let's dive into it, because the solution to this exact problem is going to completely change the way you think about genomic data. All right, so here's our roadmap for today's explainer. First, mapping the genomic landscape. Second, visualizing DNA in 2D. Third, using effective navigation markers. Fourth, hunting for sequence patterns. And finally, comparing and aligning genomes.
Okay, let's jump right into Section 1, Mapping the Genomic Landscape.
Think of the human genome like this massive, completely uncharted continent. The core problem we face with traditional DNA sequencing is that reading it is exactly like reading that single endless line of text we just talked about. It's this dense, linear string of A's, C's, T's, and G's. And because of that, it is virtually impossible for the human eye to spot overarching structural patterns or even comprehend the larger landscape of our biology.
So how do we fix this? Well, the browser takes that linear DNA and it actually wraps and stacks it. It reads from left to right and then drops down to the next line, top to bottom. It's actually a lot like the horizontal scan lines on a vintage TV set. By taking all that data and stacking it into a two-dimensional matrix, a spatial map suddenly begins to emerge right in front of you.
Now, to really understand this map, we need a super quick chemistry refresher. GC content refers to the percentage of guanine and cytosine bases in a sequence. Why does that matter? Because G and C bind together with 3 hydrogen bonds, meaning their chemical bond is actually stronger than the 2 bonds between adenine and thymine. This specific chemical makeup literally acts as the fundamental building block for the visual terrain we're about to explore.
Which brings us to Section 2, Visualizing DNA in 2D.
Let's see how that chemistry actually translates into a map you can use. Anchor your attention to that green background for a second. The intensity of that green represents the average GC bases in that specific block of DNA. And on top of that terrain, you see those colored blocks and rectangles? Those are gene annotations. They're the known landmarks of our biology, overlaid perfectly onto the structural map of the genome. If we zoom in a bit closer to the individual sequence letters, you'll notice the UI uses color contrast to help your eye immediately grasp the chemical bias of a region. Dark map regions, think black and dark blue, indicate areas high in adenine and thymine. On the flip side, bright yellow and orange instantly highlight the strong GC-rich zones. The beauty here is that you don't even have to read the letters to know the structural makeup of the DNA you're looking at. You literally just follow the colors.
Moving right along, Section 3, Using effective navigation markers.
When you're dealing with a map this massive, knowing exactly how genome markers work effectively is absolutely crucial. It's the only way to avoid getting hopelessly lost. The software uses specific built-in pins to chart your course. First, you place a bold blue circle right at your starting position. Find another interesting sequence? Boom! Drop a green circle to mark that second point of interest. And finally, a red circle wraps around the green one, so markers placed in the exact same spot don't visually obstruct each other. It's just a brilliantly effective way to calculate genomic distances and save your spots in a sea of billions of letters. To make navigation even more seamless, these markers work hand in hand with some really intuitive controls. You can just double-click anywhere to lock your zoomed-in location. If you wander a bit too far off, no problem. Just hit the jump button to instantly snap your view back to one of those saved colored markers. And if you need to travel vast genomic distances quickly, holding Shift while using your arrow keys increases your panning speed by 10 times. It makes exploring incredibly fast and efficient.
All right, Section 4, Hunting for sequence patterns.
This is where it gets incredibly cool. We're shifting from UI tools to the biological markers hidden deep within the data itself. Check out the number 171. If you go to the centromere region of a chromosome, the data usually just looks like random static. But if you alter your view width to exactly 171 bases per line, a biological marker called the scene P sequence magically aligns into a perfect repeating vertical column. It's a hidden biological structure instantly revealed just by formatting the width of our map. No way, right?
The software also lets you do brute force pattern hunting. Take this staggering number,
6062. The creator of the software ran a search for a specific 288 base repeating ALU element. In just one single section of chromosome 1, the browser located 6,062 occurrences of this exact sequence marker, just completely littering the genome. Talk about finding a needle in a haystack.
Notice how biological markers, like the telomeres at the ends of chromosomes, literally light up on the display. The browser lets you hunt using exact matches, reverse complements, or even wildcard searches. By replacing bases with a dot, you can allow for specific mismatches. This lets researchers find hidden evolutionary landmarks even if the DNA has slightly mutated over time.
Which brings us to our final stop, Section 5, Comparing and Aligning Genomes.
So, how do we compare these massive maps across entirely different species or even different chromosomes within our own bodies? Enter the dot plot. This incredible visualization tool puts one sequence, let's say the human genome, on the x-axis, and another sequence, like the chimpanzee genome, on the y-axis. By plotting the intersections, you can rapidly visualize evolutionary overlaps, as well as distinct insertions or deletions where the species diverged.
And the color coding here is a total game changer for analysis. When two sequences match exactly in a forward direction, that diagonal line is highlighted in vivid green. But if there's a reverse complement match or a sequence mismatch, like exactly where the shared DNA ends between an X and Y chromosome, the software uses a contrasting purple or magenta. You can immediately spot exactly where the differences lie. And let's see how this all builds to our final, most detailed view. By simply holding Shift and double-clicking on that macro-level DOT plot, you seamlessly zoom all the way down to a base-by-base local alignment. You can literally see the two sequences overlaid right on top of one another. It brings our journey from exploring this massive genomic continent all the way down to the microscopic individual letters full circle.
And that wraps up our explainer for today. As we've seen, transforming 3 billion linear letters into a structured, navigable 2D map unlocks a completely new way of understanding biology. By knowing how genome markers work effectively, placing those digital pins, and uncovering hidden biological sequences, the entire terrain becomes completely searchable. So I'll leave you with this thought. With these incredibly powerful visualization tools freely available out there, what new biological landmarks might you discover in your own genomic exploration? Thanks so much for joining me on this deep dive.